Okay, so in this video we are going to be discussing about the anatomy of the kidney, in which we're going to be starting with the blood supply. So kidney receives the blood supply from the abdominal aorta, right? Both of the vessels. Now for the vein part, let's just discuss the artery. It is going to first divide into segmental arteries because there are different segments in the kidney, and then these segmental arteries are going to divide into the interlobular artery, which is going to be dividing into afferent arterioles, then glomerulus, देन efferent और फिर वो पेरीट्यूबुलर कैपिलरीज में डिवाइड हो जाती है और फिर ये इवेंचुअली ज्वाइन होकर इंटरलॉबुलर वेन बना देती है. अब वेन्स में हमने क्या बात करनी थी कि जो लेफ्ट रीनल वेन होती है उसमें दो वेसल्स और ड्रेन कर रही होती हैं, एक लेफ्ट सुप्रा रीनल और एक लेफ्ट गोनाडल. लेफ्ट ओवेरियन भी, टेस्टिकुलर भी. इसीलिए जब लेफ्ट रीनल सेल कार्सिनोमा होता है तो उसके चांसेस ऑफ गेट डेवलपिंग इनटू द लेफ्ट वेरिकोसील इज़ वेरी हाई. बिकॉज वाई, जब यहाँ पे वो इन्फिल्ट्रेट कर जाएगी रीनल वेन में तो आप ऑफकोर्स आपके पास जो गोनाडल वेन होगी वो तो ड्रेन नहीं कर सकती, तो आपके पास पीछे पूलिंग ऑफ ब्लड होना शुरू हो जाएगी लीडिंग टू वेरिकोसील. ठीक है. अब उसके बाद वी आर गोना बी डिस्कसिंग अबाउट दैट के जो कॉर्टेक्स होता है मेन हमारे पास बाहर वाला पार्ट है, मेडुला जो अंदर वाला पार्ट होता है, कॉर्टेक्स को मेनली ऑक्सीजनेटेड ब्लड जाता है और कम ब्लड जाता है टू द मेडुला. तो जब भी कभी इस्कीमिया होगा या कम ब्लड जाएगा किडनी को तो जो मेडुला पार्ट होगा उसमें हाइपॉक्सिक कंडीशंस जनरेट होना ज़्यादा मतलब पॉसिबिलिटी होती है, तो वो ज़्यादा जल्दी डैमेज होता है. सो व्हेनेवर देयर कंडीशंस लाइक एक्यूट ट्यूबुलर नेक्रोसिस व्हेन ब्लड सप्लाई टू द किडनी इज गोना रिड्यूस, दैट मीन्स दैट योर मेडुला इज द फर्स्ट वन टू get affected. So for the glomerulus anatomy, it is very important to understand this, that the blood is going to enter through the afferent arterioles. Now these afferent arterioles have these modified smooth muscles called as the juxtaglomerular cells, which are going to release renin. That is associated with the sympathetic supply, that either renin is going to be released or not. Now you can also see these macular denser cells which are present in the distal convoluted tubule as sodium sensors. Now if the sodium content is low in the blood, it is going to cause renin release, and then more sodium reabsorption is going to occur. But if there's going to be too much of the sodium, it's going to cause constriction of afferent arteriole, which is going to increase the GFR. So more of the sodium is going to get excreted out of the body into the urine. Alongside of that, you can see the mesangial cells. These are said to be the supporting cells or the cleaner cells of the kidney, and they also have a role for the macrophages. They basically engulf bacteria and also remove the debris. They modify glomerular filtration rate by releasing a lot of cytokines, and then they also help to maintain the structure. For GFR barrier, whenever we talk about glomerular filtration rate, there's this barrier that we need to talk about. So there is going to be the basement membrane. Okay, and then these podocyte cells are going to form the food processes of podocytes. So there are three components of it: food processes of podocyte, the basement membrane of the capillaries, and then the fenestrated epicapillary endothelium. So you can see that this is your capillary, this is the fenestrated endothelium of the capillary. So one, this, second is the basement membrane of it, third is going to be the food processes of the podocyte. So this is your glomerular filtration rate. So that's it for the anatomy of the kidney. And no, actually there's another part of it, which is the course of ureter that you need to know. For the course of ureter, you need to know this, that it basically travels under the gonadal arteries, testes and ovarian artery, and then it passes through the common iliac artery. The common iliac artery is going to divide into, for example, abdominal aorta divides into right and left common iliac artery, and then external and internal iliac artery, right? So this ureter is going to pass above the common iliac artery, and it is going to be below the gonadal arteries and the uterine artery, and in males beneath the vas deferens. For the procedures of hysterectomy, the ligation of when uterine and ovarian vessels is being done, it is very important to prevent the ureter from getting ligated in this process. Three very common points in which ureter is going to get obstructed. One was the pelvic and ureteric junction. Second is when it's going to enter into the pelvis. Third is when the ureter is going to enter into the bladder itself. Sorry for my drawing and handwriting. So yeah, that's it for the anatomy, and then we're going to be moving on to physiology.